All right, my talk today is on the development of the VARITAS database and pipeline for rapid microbial testing in biomanufacturing. My name is Tyler Laird. I'm a bioinformatician in the Complex Microbial Systems Group at the National Institute of Standards and Technology, or NIST. So an outline of my talk today, I'll give some background on NIST and the Rapid Microbial Testing Methods Consortium that we run, as well as information on the VARITAS database and pipeline creation, and then subsequent to that, pipeline validation analyses. So to start off with some background on who or what exactly is NIST, we are the National Institute of Standards and Technology, which is a non-regulatory federal agency within the United States Department of Commerce. NIST's mission is to promote U.S. innovation and industrial competitiveness by advancing measurement science, standards, and technology. I am part of the Complex Microbial Systems Group, which focuses on microbial metrology or measurements related to microbiology. We have two main areas that we focus on, one being the microbiome or measurements related to complex microbial communities and how that can be related to engineering microbial communities, as well as biosurveillance, which focuses on pathogen detection. And that's kind of where the VARITAS database and pipeline fit in. And so members of the Complex Microbial Systems Group, along with other bioscience-related groups at NIST, got together to form the NIST-led Rapid Microbial Testing Methods Consortium, or RMTM consortium for short. And this stems from the fact that the safety and quality of advanced therapies, including cell therapy, gene therapy, and tissue-engineered products, must be maintained prior to patient administration. And the current culture-based compendial methods typically take weeks. And so that's diagrammed on the bottom here, where you have a cell therapy product sample being taken and inoculated into two different types of broth culture media. And then you have to wait up to 14 days to assess for turbidity, which tells you whether or not the product is contaminated. And so alternative rapid microbial testing methods are really needed in this area, especially for biotherapeutics, which need a rapid turnaround time to get back to the patient. So the RMTM consortium was founded in 2020, and it brings together experts from industry, academia, and government to address RMTM needs. And there are a wide array of rapid microbial testing methods, but one particular focus that we're working on is NGS-based sterility testing. And so in 2024, through the RMTM consortium, NIST, along with the company EasyBiome, began development of a shotgun metagenomics database and pipeline for sterility testing. And so the idea here would be taking a cell therapy product sample, performing DNA extraction, subsequent sequencing, and then bioinformatics analysis to then get a readout of whether a product is contaminated or not. And the result of this collaboration was the VARITAS database and pipeline. VARITAS, which stands for Validated Reference for Industrial Testing and Sterility. And we took a variety of different lists of organisms that we included as input taxa in this particular database. So one being a minimal target organism list, which includes organisms that have been identified in the literature as contaminants of cell therapy products, such as hematopoietic stem cell transplant products, an environmental organism list, which includes organisms that have been found as pharmaceutical contaminants or within the pharmaceutical production environment. And we also included several spike-in species, which are not related to sterility testing at all, but if you spike one of these organisms in to a sample, they can be used as internal controls in order to validate a particular workflow or address particular biases in a metagenomics pipeline. And so taking all of these lists of organisms to create the database, the goals were to include only validated species in the database, so no genome species or unclassified organisms, and to include one genome per each species. And the inclusion preference tiers are listed below that. So starting off, ideally we'd have a reliable type strain for each one of these genomes. If that wasn't available, we moved on to a NCBI reference genome, followed by a complete genome or a scaffold assembly. And if none of those were available, a contig assembly. Then taking all of those genomes, they were then subject to additional validation and ANI-based grouping. Either these particular genomes were grouped together if they had an ANI value above 95%. So as was the case for Escherichia coli and Shigella species, or as the example here shows, certain genomes were separated out into multiple groups, even if their names were the same. So that was Clostridium botulinum separated out because their ANI values were below 95% for certain comparisons. And this ultimately resulted in the VARITAS database, which contained genomes from 2,543 bacterial species and 654 fungal genomes. So now that I've talked about the database itself, I'll go more into the bioinformatic pipeline. This can be kind of broken up into two main chunks. The first being pre-processing, where we're taking the input FASTQ files and subjecting them to quality control with FASTP, the complexity filtering with BB tools, and then host read removal with hostile. And then the main VARITAS workflow, which incorporates the core genes from the entire VARITAS database that I mentioned. So we have 81 bacterial and 61 fungal core genes that are extracted from the database genomes if they're available in those genomes. And then the reads of a sample are classified using Kraken2 against those core genes, and then subsequently mapped with Bowtie2. And then SAMtools is used as for some file manipulations to subsequently create an output that indicates what taxa were detected and the level or the percent of core genes that were detected for each taxa. So now that I've talked about the pipeline and database creation, I'll move on to the pipeline validation. In this first particular analysis, looking at in silico simulated reads of compendial organisms. So first off, what exactly are compendial organisms? Well, they're microbial strains that must be used as challenge organisms in quality control and sterility assurance testing. So there's these different pharmacopoeias that different countries have that have been harmonized to validate sterility testing, and they include these particular organisms listed in the table below. So these are the genome names that we got to simulate from, and on the right-hand side, the organism features that there's a variety of different classes of organisms here, gram-positive and gram-negative bacteria, as well as aerobic bacteria, anaerobic bacteria, and two fungi representatives, one being a yeast and another a mold. And so we obtained these genomes and then simulated varying amounts of Illumina reads with the ART software. And this is one of the outputs of this analysis. Really looking at the precision, which depends on the taxonomic level and the organisms assessed with the VARITAS workflow. And so on the left-hand side, you can see a plot where we have the number of simulated reads on the x-axis and the core gene fraction on the y-axis. And this is an example for the Staphylococcus aureus simulated read experiment. And Staphylococcus aureus is the true positive organism, in this case, represented by this purple-blue bar color. And so as the number of simulated reads increases, the core gene fraction for the true positive Staphylococcus aureus increases to 1, as you would expect. There's also some false positives, other Staphylococcus species that start popping up around 10,000 reads and above. And so, one thing to note is that these can be reduced if there's a core gene fraction threshold implemented, but that really necessitates that you have a sufficient number of reads. to classify. So for instance, if you set it at 0.4, you can cut off most of these false positives from being detected. However, you would need to have at least 1,500 or more true positive reads in order to implement that. We can look at this table over here, which breaks down the all of the simulations that we did for these compendial organisms, specifically looking at the 100 million or the 1 million read simulation analysis, and looking at the precision, which is the number of true positives over the number of true positives plus false positives. And we looked at the species and the genus level. And what you can note here is that the precision at the species level is variable. So some of these organisms were able to be detected with high precision, whereas for Bacillus spezzini and Candida albicans, the species level precision was relatively lower. However, at the genus level, precision was 1 across the board. And so the vast majority of reads are correctly identified at the genus level using the VARITAS pipeline. We can also look at some other validation experiments where we're comparing the VARITAS pipeline with other tools and databases that are available. So in this case, we put those 1 million reads through VARITAS along with Kraken2 Bracken, Centrifuge, and MOTUs with default databases that are available for those. And again, looked at precision as well as another metric, which is read utilization, or the number of reads classified over the total number of reads. One thing to note is that the VARITAS database contains the relevant taxa in this scenario. So certain tools and databases like Centrifuge and MOTUs don't represent fungi. And so Candida albicans and Aspergillus brasiliensis aren't even able to be detected. Another thing to note is that the VARITAS pipeline, like MOTUs, is a marker gene approach, but VARITAS has a much higher read utilization compared to MOTUs, and that's because VARITAS is using 81 bacterial or 61 fungal core genes compared to MOTUs, which uses a lower amount. And the VARITAS pipeline really minimizes the false positive problem. So another thing to highlight in this table is the number of unique false positive taxa, which is indicated in the top of each one of these panels. And you'll note for VARITAS, it's all 0 except for Aspergillus brasiliensis, where there were 5 unique false positive taxa detected. And compare that to the non-marker-based pipelines and databases like Kraken2 and Bracken and Centrifuge, there were tens to hundreds of unique false positive taxa detected. One final pipeline validation analysis we can look at is the spike-in data that's available on the Sequence Read archive. And so this data was uploaded from a study, a systematic sequencing-based approach for microbial contaminant detection and functional inference by Park et al. And they essentially took these compendial organisms, grew them up, and spiked them into mesenchymal stem cell cultures at either 1,100 CFUs or 60 CFUs. And this table shows the breakdown of those reads that were detected and classified to these taxa at the species and genus level for VARITAS, which is boxed in red, and also the number of genus reads that were classified according to the Park et al. Analysis and the particular pipeline they used. And so one thing to note is that at the 1,100 CFU level, most of the organisms get detected. So 5 out of 6 at the genus level, 4 out of 6 at the species level. However, when you get to the 60 CFU level, there's only 2 organisms detected at the genus level for VARITAS. And this really shows that, one limitation of this pipeline is, on low abundant samples, especially because it's utilizing only the core genes for classification. So this gets me to the conclusions for my talk today. So the VARITAS pipeline has high precision at the genus level. It's variable at the species level, but very good and reduces the number of false positive taxa detected compared to other pipelines. One challenge is dealing with the low microbial abundance data sets, like I just showed with the Park et al. Analysis of their data set. And this requires refining wet lab workflows in combination with bioinformatic workflows. So definitely making sure that host read removal is optimized. So future work for the VARITAS database and pipeline, we are testing additional data sets as part of an in silico interlab study. NIST, which I work for, does a lot of these interlab studies where we compare pipelines and workflows from various labs, and we're currently recruiting participants. So if this sounds like something you're interested in to help advance the use of NGS in the for sterility testing of biotherapeutic products, definitely contact me if you'd like to join. As part of the actual pipeline, we'd like to integrate alternative NGS strategies, including long read shotgun metagenomics, amplicon sequencing, and RNA sequencing. And I'd like to acknowledge everyone who worked on this project. So Michael Netherland, Seon Choi, Nur Hasan, Stephanie Servitus, and Scott Jackson for being instrumental in creating this database and pipeline. Thanks for listening.